Today we are going to start looking at photosynthesis. In this lesson, it is just an overview, so you're going to see all of the things involved, but just little snippets. So some background information and just an overview of the process. We then dive deeper into the light reactions, so the photosynthesis maybe we're familiar with. And there's two different lessons for that. And then we will get into the third part, which is light independent reaction. So let's look at some background information and a reminder of some terms that will be helpful. Photoautotrophs use the organic molecules that they create, so glucose, from inorganic raw materials like carbon dioxide, for their own activities and as organic building blocks. And we're going to look at what more is produced other than just glucose. They serve as food sources for consumers like ourselves. They use some of the organic material for energy and for building blocks for their own bodies. Recall that producers and consumers eventually become energy and building material for decomposers. So we all die and decay. It's the circle of life. Lastly, I want you to remember the second law of thermodynamics. Entropy, so the randomness, if you will, increases with every energy transformation or chemical reaction. So useful energy is lost at each stage of the cycle. Free energy needs to be inputted from the sun. And now let's take a moment to thank the plants for photosynthesis so we get to breathe and eat. In the light-dependent cycle, we have light energy being captured by pigment molecules such as chlorophyll. Chlorophyll we are familiar with as the green pigment found in plants. The sunlight is used to create NADPH, and we can read nicotinamide adenine dinucleotide phosphate. So this looks a lot like NADH, which we see in glycolysis and cellular respiration. So this is a similar product. It is being used to carry electrons to an electron transport chain. We're also getting the production of ATP, which will be used a little bit later on. Interestingly, we have water being split in these reactions, and 2 electrons are taken, and they get passed along an electron transport chain, similar to what we see in the mitochondria. And the final destination is the reduction of NADP plus to NADPH. And again, this is an electron carrier molecule. At the same time, we're getting the establishment of a proton gradient across the chloroplast membrane. Again, similar to cellular respiration in the mitochondria. At the same time, we have oxygen being produced as a waste product. And again, we have water being used, and when it is split, we're getting that release of oxygen. And again, we have energy being stored as ATP. In the Calvin cycle, we are using that NADPH and the ATP that was produced during the light-dependent reactions. Here we get carbon dioxide being converted to a simple 3-carbon carbohydrate called G3P, if you remember from glycolysis. This G3P is used to make other larger molecules like glucose, or we can get lipids and proteins as well. All organic molecules of plants are an indirect or a direct product of photosynthesis. And make sure you record the overall photosynthesis reaction. We have carbon dioxide and water reacting to produce glucose and oxygen. And this is done with light energy. So we have here the structure of a chloroplast, and in a single cell we have a lot of chloroplasts. So don't be mistaken by thinking there's just one chloroplast doing all of the work. Both stages of photosynthesis, the light-dependent and light-independent reactions, occur in the chloroplast. The chloroplast has 3 membranes, and it is special because it creates different compartments. So we have the outer membrane, and we have our inner membrane, and we have our intermembrane space that is between those 2 membranes. In our next slide, so let's dissect this image of a leaf. So here is a leaf right here. And we see these photosynthetic cells. And so zooming in on one cell, we have a large vacuole, we have the nucleus, and a whole bunch of chloroplasts. Looking at one chloroplast, we see our inner and outer membrane, and we have the intermembrane space between them. And then we have the space that is inside. Now the aqueous environment is called the stroma. And then we have another membrane that is called the thylakoid membrane. So this membrane is creating the thylakoid stacks. So each one of these is a thylakoid, and when they're stacked, they're either called a thylakoid stack or a grana. The thylakoid lumen is the space within that thylakoid. So a bunch of terminology for you to get familiar with, because with photosynthesis, we're going to talk a lot about the stroma and the thylakoid lumen. So let's talk about what happens, some hypothetical situations from chemistry. What happens when a photon of light is absorbed by an atom? What's going to happen if we have our photon of light? If it hits an electron, this electron can get energized and jump to the next excited state, so a higher energy level. So when the electron is not hit by a photon of light, it is orbiting in its ground state, so it's low energy level, ground state. And then if it gets hit by a photon of light, it can jump to the next energy level. This becomes important in the next couple of slides. So let's look at what could happen if that electron gets hit by a photon and jumps to the next higher energy level. So this is our atom, and an electron got hit by a photon of light and it jumped to its excited state. 3 things can happen. So let's look at outcome number 1, which is kind of boring. It just returns to its ground state. It releases energy as thermal energy or as fluorescence. So not much happens, I would say. Our next outcome would be that the energy of the electron is transferred to an electron in a neighboring pigment molecule. So what does that mean? It's going to release energy and activate a different electron in an atom that is very, very close to it, in a different pigment molecule. And it would then go back to its ground state. So this one goes back to normal, but it activated a nearby pigment molecule, a nearby atom. And we'll talk about, we'll see a diagram of what this looks like in use. The third outcome is that that electron actually jumps to another atom and it gets accepted by another molecule. And again, we'll see what this looks like. So in our 3 different states, we have essentially nothing happening except heat being released. This one, energy gets transferred to another pigment molecule. And in our third case, the entire electron jumps to another molecule. So thermal energy released to activate another electron, and actual movement of the electron. And in this third case, we have a definition. So this electron is jumping to another molecule, and it's called a primary acceptor. So this is the molecule that is capable of accepting electrons and becoming reduced during photosynthesis. Remember, it's being reduced because its charge is being reduced. It's becoming negative. So what happens to light energy? So remember, we get 100% light energy from the sun. And again, we're just looking at visible energy. We see 11% gets reflected right off the leaf. We see another 2% that gets emitted as fluorescence. About 11% goes right through the leaf and does nothing. 20% gets released as thermal energy. So this was, this 20% and this 2% was outcome number 1. And then we get 56% approximately being used in photosynthesis. And that's actually the use of outcome number 2 and 3, which we will talk about. Some more definitions. So chlorophylls are the major photosynthetic pigments in plants, and I feel this is probably a definition we're kind of familiar with. It's also found in green algae and cyanobacteria, so very common pigment. Most abundant are chlorophyll A and chlorophyll B, so there's different types of chlorophyll. Most abundant are A and B. We also have bacteriochlorophylls. These are similar to plant chlorophylls, but they're found in photosynthetic prokaryotes other than cyanobacteria. And then we have carotenoids. These are a second major group of photosynthetic pigments. We are familiar with maybe them being orange, as found in carrots. So we have a costume change. The previous recording didn't work, so here we are on another day. So let's look at one of these action centers, if you will, that would be in a photosystem of our chloroplast. And we'll talk a little bit more about that in a bit. So we have, let's label all these guys. We have our antenna chlorophyll molecules. And again, these are bound to a protein, keeping them in the thylakoid membrane. We have our carotenoids and other accessory pigments. We have our chlorophyll A, which is going to end up giving its electron to the primary electron acceptor. So we have a photon of light that hits one of the antenna chlorophyll molecules, and we're going to watch this transfer of energy. So energy, not an electron, is transferred from one chlorophyll molecule to another. And then ultimately, we get our chlorophyll A electron activated to the point where it is going to jump to a primary electron acceptor molecule. So the primary electron acceptor gets reduced, again, because it is taking an electron. So here is the diagram that is in your textbook. And so I just wanted to animate it so we can follow along. Again, a photon of light comes in, and we see this transfer of energy from chlorophyll molecules until we ultimately get to our chlorophyll A, which will donate its electron to the primary electron acceptor. So again, we have some definitions to address. So chlorophyll A here becomes oxidized because it's losing an electron. And as mentioned in the animation, the primary electron acceptor gets reduced because it is taking an electron. Chlorophyll B and carotenoids are accessory pigments. So they get energized, they transfer their excited energy to chlorophyll A. So this is like an assist in hockey. And then we have the antenna complex. So this is the entire cluster of light absorbing pigments. And again, these are embedded in proteins that are embedded in the thylakoid membrane. They capture and transfer energy to special chlorophyll A molecules in this reaction center. It's important to know that pigment molecules do not absorb all wavelengths of light. So similar to any other object that we see, for example, this mug appears yellow because it does not absorb yellow wavelengths of light. So it reflects yellow, so the yellow hits our eyes and we see this as yellow. The same is true for chlorophyll pigments. So chlorophyll A, for example, does not absorb green and yellow wavelengths. So this is why leaves appear green. So we have an absorption spectrum. So it looks a lot like a mass spec that we might have looked at in grade 9 with the stars in the sun. So this is a plot of the absorption of light as a function of wavelength. So here we see the absorption of light percentage, so how much light gets absorbed based on the wavelength of light. And again, this is read from 400 nanometers, and then it's plotted 700 nanometers. So we use a spectrophotometer to analyze a sample of a pigment. And chlorophyll A strongly absorbs blue and red light. So let's look here. We have chlorophyll B right here, carotenoids, and chlorophyll A. So if we follow along chlorophyll A, it's the solid line. It is peaking in the blue-purple area, and again in the red. So it's going to be absorbing colors there. So it's going to be reflecting the green and yellow. Similarly, we can look at chlorophyll B. So it peaks in the greenish area and again in the orange. So we would see these as green and yellow and maybe even red. Carotenoids absorb the purple, blue, greenish colors, and that's why we see plants that are high in carotenoids as the orange and red and yellow. So what's interesting is the vivid colors in fall, we have a degradation of chlorophyll A, and so we get to see other pigments. So that's how we see the change in color of leaves. So similar to an action or an absorption spectrum, we have an action spectrum. So this is a plot of the effectiveness of light of various wavelengths during photosynthesis. So again, they use a suspension of chloroplasts or algae cells and measure the amount of oxygen produced, which is shows that photosynthesis is occurring. So let's look at this graph. So we have the rate of oxygen released in photosynthesis, and we have our different wavelengths of light. So we have, showing here, we have a high production of oxygen when the chloroplasts are given blue light, not much when it is given the green and yellow light, and again we have another peak with the red light. And that's because this is the light that can be absorbed by these pigments. So the pigments can't absorb green or yellow light, so it's not going to be very effective when given green or yellow light. So you're going to have a low rate of oxygen production. Okay, so let's quickly look at the photosystems, and then that is a wrap for this introductory lesson. So we have two different photosystems, but we are just going to talk about a generic photosystem structure to begin with. So the pigments shown here, these little green spheres, these are pigment molecules, and they are bound to proteins in the thylakoid membrane. So we have a thylakoid membrane here. These are large complexes, so light harvesting complexes, and they have 250 to 400 pigments each. So a little underrepresented in these diagrams. And they are said to be surrounding a central reaction center. So this is the reaction center. And we'll see in our next lesson what that looks like. So what kind of reaction is happening. You can imagine it involves the transfer of an electron. The center is made of a small number of proteins, each bound to a pair of specialized chlorophyll A molecules. So this is your chlorophyll A molecule. And we have our primary electron acceptor. So this is where the magic is happening in this action center. So light energy that's absorbed from the sun by this antenna complex ends up getting transferred. So we see a movement of energy, and it ultimately ends up at this special chlorophyll A molecule. Light is converted to chemical energy when this reaction center chlorophyll donates an electron to this primary electron acceptor. This electron gets passed along an electron transport chain. So we have, like I said, two photosystems. Photosystem 1 actually comes first, but it's called photosystem 2. I didn't make it up, guys. I'm sorry. This reaction center contains chlorophyll A called P680. This absorbs wavelengths of light best at 680 nanometers. We then have photosystem 1. So it was discovered first, so that's why it's called photosystem 1. But it comes second in the process of photosynthesis. Its reaction center is very, very similar, except it has P700. So P700 absorbs wavelengths of light best at 700 nanometers. And again, this is a chlorophyll A molecule. And again, when it gets activated, it'll donate an electron to this primary acceptor here. We will look at all of the specifics in our next lessons. Can you guess what this is based on its structure and our knowledge from previous lessons? I'm not telling you. So in summary, we have photosystems, like one shown here. They trap photons of light from the sun. They use energy passing from chlorophyll to chlorophyll and ultimately activate chlorophyll A in this action center. The chlorophyll A molecule is then oxidized as it donates an electron to the primary electron acceptor. And we're going to see, again, all of the specifics of these reactions in the upcoming lessons.